Hello and welcome to another online lecture from the University of Washington's Synthetic Biology course. So today we're going to talk about primer design. To start off, imagine you have a plasmid of DNA. And as we know, a plasmid is just a circular loop of DNA that is common in bacteria. Also imagine that we are particularly interested in this segment of DNA denoted in red. Well, how would we go about isolating that segment of DNA from the other thousands of base pairs in the entire plasmid? Well, we're going to need to do something called primer design and use a tool known as PCR. If you are unfamiliar with PCR, go ahead and scroll down to the description section where a hyperlink is posted that will direct you to a video that will explain PCR further. So, assuming that you are familiar with PCR, we'll continue forward. What I've drawn down here is a zoomed in view of the region of DNA that we are actually interested in. And as you remember, each of these letters dictates a different base that is available in DNA. So we have G's, C's, A's, and T's. And as you might remember, G's bond with C's, and A's bond with T's. And G's actually bond with C's with 3 hydrogen bonds, and A's bond with T's with 2 hydrogen bonds. So a G-C combination is actually a stronger bond than an A-T combination. That will be important later on as I explain something. So, in order to isolate the region of DNA that we are actually interested in, we need to design primers that will dictate that region. We do that by designing one forward primer and one reverse primer. I will explain this further in the expanded view of our segment of DNA below. So as you know, DNA is double-stranded and written classically in the 5' to 3' direction. So that means that the complementary strand, or this strand down here below, is written in 3' or to 5', which means that it is actually written backwards to standard convention. So if I was going to design a primer that worked in the forward direction, as shown above, this forward direction here I'll mark it for you as forward. This one is forward,